This is me and Molly's presentation on DNA replication.
The cell cycle consists of multiple phases: G1 growth, S phase, G2 interphase, mitotic cell cycle, PMAT, and cytokinesis. DNA replication occurs in the S phase of the cell cycle.
DNA reads 3 to 5 but is added 5 to 3. 5' refers to the amount of carbon that is on the end of the nucleotide. The 3' is a third carbon with a hydroxyl group attached. DNA runs anti-parallel, meaning this top strand runs 5 to 3 and the bottom strand runs 3 to 5.
DNA has 4 base pairs: adenine, thymine, guanine, and cytosine. These are complementary base pairs, meaning that adenine and thymine will always bind together, and cytosine and guanine will always bind together too. There are 2 different categories of nucleotides: purines and pyrimidines. Purines have a 2-ring structure versus a pyrimidine single-string structure. Adenine and guanine are purines, and thymine and cytosine are pyrimidines. Purines and pyrimidines always bind together. These nucleotides are held together by weak but stable hydrogen bonds.
A chromatid is one of the 2 identical halves of a chromosome. A sister chromatid is 2 identical copies of a single replicated chromosome. The sister chromatids are what is formed during DNA replication.
The origin of replication is a specific non-random DNA sequence that denotes the area of active replication, acting as a binding site for proteins like helicase. In prokaryotes, there's only one origin of replication, whilst in eukaryotes there are multiple sites. In the Ori, helicase will begin to unzip and separate the strands of DNA, creating 2 DNA strands and a replication fork. The separated strands provide a new template for creating the new DNA.
Helicase is the enzyme that unzips the DNA. This forms a replication fork. The DNA must be held apart by single-stranded binding proteins because the DNA will want to bond back together. DNA gyrase is used upstream of the replication fork to prevent any supercoiling of the DNA. Supercoiling is the excessive twisting of DNA, which can be detrimental to the genetic material if too extreme.
To start the actual process of DNA replication, after helicase has separated the strands, primase is needed. Primase makes small RNA pieces called primers. These mark the starting point for making a new strand. This helps with the DNA polymerase as it can't add nucleotides without a primer being present. It's important to note that the location on DNA is specific to where the primer binds to. They usually recognize a specific trinucleotide sequence.
DNA polymerase is the enzyme responsible for adding free-floating nucleotides onto the primers, creating the new strand. The beta clamps are ring-shaped proteins that surround the single strand of DNA to ensure the DNA polymerase continues to adding nucleotides. The beta clamp loader is a protein that opens the beta clamp to allow it to surround the single strand of DNA so that it can perform its function.
The continuous synthesis strand, also known as the leading strand, is 5' to 3' strand because DNA polymerase can continually add in that direction. Discontinuous occurs on the lagging strand, which runs 3' to 5'. Since DNA polymerase cannot add continually in this direction, new primers must be added, and then DNA polymerase must go back and add the nucleotides.
Since DNA polymerase can only add 5 to 3, the 3 to 5 strand must be added discontinuously. This occurs on the lagging parent strand. It's completed in short segments of DNA nucleotides called Okazaki fragments. Each fragment begins with an RNA primer, and primer is added as the DNA is being unzipped ahead of the 5' end of the lagging strand. DNA polymerase then adds the Okazaki fragments to the primers. This fills the gap as the helix is opened further. After the DNA polymerase replaces and removes the RNA primers with DNA nucleotides, it's then linked together by ligase to ensure bonding and to form the lagging strand during replication.
DNA polymerase 3 is the enzyme that adds nucleotides to primers in prokaryotes, for example, E. coli. DNA polymerase 1 is responsible for the removing of the RNA primers and replacing them with DNA nucleotides in prokaryotes.
DNA ligase is the enzyme that glues the semi-conservative strands together. Ligase does this by forming the phosphodiester bonds.
The semi-conservative region is an area where each new DNA molecule consists of one original strand and one newly synthesized strand. These original strands can also be called parent strands. The parent strands act as templates for the synthesis of a new strand, the daughter strand. After replication, the parent strand is now paired with the new daughter strand.